Okay, everybody, I'd like to welcome Jessica Lee. Jessica heads up the biostatistics program at Fred Hutch and is also affiliated with the University of Washington. And today she'll be telling us about question inferring causal gene regulation from Perturb-seq.
Thank you. Thank you, Ben. Thank you, Yogesh, for the invitation. And this is my first time talking about this problem because I would say my group is still quite new to the Perturb-seq field, even though we have been working on single-cell RNA-seq for quite a while. But this is something my postdocs have delved into, and we just noticed that this is something like people discuss in a very different ways in different method papers for inferring causal gene regulation. Essentially, if you use Perturb-seq to perturb certain genes, you are interested in their downstream genes. What are the genes affected by these perturbed genes? But how can we make sure that the causal statement is accurate? And to make a causal statement, we need to answer this question first, which is essential in any causal inference problems. That is, are the cells randomly perturbed? Do we actually have a random experiment? So I will just be brief since Tom already given very good review about the technologies, and we also had yesterday. So Perturb-seq is a combination of using CRISPR-based genetic perturbation to change our gene expression, followed by single-cell RNA-seq as the readout. The key is that now we have simultaneous measurements of thousands of genes, and we also have many perturbations happened in one experiment. So that's why we're using the high throughput of millions of individual cells to capture many perturbations and measure their transcriptional consequences. Regarding the perturbation part, we can have CRISPR for doing gene knockout. This is actually breaking DNAs, and this is the first approach, Perturb-seq. And the most common approach, as Tom mentioned, is CRISPR-I, interference, which is essentially repression. We work on the repressing RNA expression without altering DNA. And he also mentioned CRISPR-A for activation, and this is the most challenging one. And we want to activate certain genes, then we measure the consequence. So this is more like overexpression. So CRISPR-I is where most data have been generated from. And for CRISPR-I, this is also the paper, 2022 paper, Tom mentioned his talk, is essentially applying, you can see that you have many, many cells to begin with. You need to get those single guided RNA into those cells to repress certain genes, and you will measure the guide RNAs again together with the other genes expression in millions of cells. So essentially, the data you get out of the experiment are two matrices. So you will have a gRNA assignment matrix, and you will also have a gene expression matrix measured in UMI counts. And this is from single-cell RNA-seq data. So essentially, you want to learn from the gRNA assignment matrix in which cells you have which guide RNAs targeting which genes. And then this can give you the red colors in the single-cell RNA-seq matrix. So you know that in cell 1 and cell 2, gene 1 is perturbed, and in cell 3, gene 2 is perturbed. But as you can see that different guide RNAs, even though they target the same gene 1, they may give you different efficiency. So that's why gene 1's count in cell 1 is not completely down to 0, right? And gene 2 in cell 3's count is also 2 instead of 0. So this raises a very interesting question. If you want to do a causal research, like which gene caused which gene, in this it's called directed acyclic graph. It's a jargon used in statistics and any causal research, DAC, short as DAC. You are interested in how X causes Y. And in our context, X is a perturbed gene. Y is a potential downstream gene. You're interested in the potential causal effect here. But we also need to understand other variables that may affect your inference on this causal relationship. Because if you don't know anything about other variables, the simplest answer to study the causal effect is simply a correlation analysis. You check the correlation between X and Y, then how strong that is, it may indicate the causal effect. However, confounder is a very important thing to keep in mind when you do any causal argument, because you as a confounder variable, if it causes both X and Y, then even though X doesn't cause Y, you will make X and Y correlated. So that's why you cannot just infer correlation between X and Y as a causal effect if you don't know anything about confounders. A more tricky thing is something called collider, which is a consequence of both X and Y. So if X causes C, Y causes C, but you misconsider C as a confounder, then by conditioning the correlation between X and Y on C, you will actually introduce spurious correlation between X and Y. So that's why for anything that's beyond X and Y, the first question to ask is, are they confounders? Are they colliders? So this is something I want to specifically raise as an open question for this Perturb-seq gene regulation analysis. Okay, so I give the overview. Now I'll be more specific. So X is the perturbed gene. But how do we measure X? There's also two ways. We can simply use the guide RNA assignment matrix to see if the guide RNA is in a cell, we think just this corresponding gene is perturbed. So in this case, you can use a binary variable to indicate guide RNA presence, but we know that guide RNA's presence doesn't mean the gene is effectively perturbed, right? Because the efficiency is unknown. So that's why if your X is just a guide RNA indicator binary, it's called intention to treat in the causal literature, which means that you intend to treat the cell, but whether it's treated or not, it's still unknown. Or you can use the measurement of the perturbed gene in the single cell RNA count matrix by treating that as a continuous treatment variable. So essentially, you want to know how much, to what degree is this gene X expression downgraded or knocked down. So if you're using this, then it's a different type of treatment. Essentially, this is called as treated instead of intention to treat. And the confounder is likely important here because if guide RNA enters a cell, to what degree does it alter the target gene by perturbation may depend on something like the cell state. For example, the cell cycle phase, which may have different cells in different phases be more vulnerable or less vulnerable to the perturbation. And this cell state can also be a factor that dictates the gene expression of other genes. So if we believe cell state can affect both perturbation efficiency and baseline gene expression of other genes, then this cell state can be considered a confounder because it satisfies this confounder definition. But the challenge here is that we don't have the pre-perturbation information about gene expression in the perturbed cells. If we only do a single time point experiment, we only measure gene expression after perturbation in the perturbed cells. So we don't know what's before, right? That is the fundamental challenge here. Okay, how about collider? Do we have colliders in this case or not? That's another question. One way to think about this, as collider needs to be a consequence of both X and Y, maybe this is a gene that is downstream of the perturbed gene and this Y gene. So if that's the case, then for such genes as a collider, defined in the collider sense, should not be used to help us find the confounder. So this is another thing we don't see much discussion in the literature. So this is very dangerous. If you treat a collider as a confounder and then you condition on the confounder do X and Y association analysis, you can have spurious association. Okay, so the questions from this causal DAC is, one, how can we learn confounders from data? Second, how can we ensure that the learned confounders are not colliders? Yes.
So I am curious how much of what is going to follow rests on the assumption that we're actually working with a DAG.
Yeah.
Yes. So can you, so I don't have to wait, and do you know how much do I care? Like, are you really strongly assuming
that it's acyclic?
Yeah.
Cyclic, yeah.
How much of what you say?
Yeah.
Do you, so basically if you want to.
I think that the actual biological system definitely has cycles everywhere.
That's right, yeah.
So is that going to be a problem for things that follow, or are you really strongly?
Exactly. So my best, okay, I'll try to answer the question. So the DAG is of course an assumption, You need the DAC to make causal statement from X to Y. That is something we need. But U and C do not necessarily need to be single genes. So it can be something it can be an abstraction of something. But essentially, if you want to make the statement that, okay, by perturbing X, this is the effect I see in Y. If you're trying to make that effect causal, some discussion about whether you have a counter for confounders or what they are need to be made transparent.
But
That's
if
the
Y
point.
affects X even,
But
many times there's a feedback of some kind, I think is the point. You could still have causality, but it's just hard.
Yeah. So that I think this is related to what I will talk later, which is about the data we need. Because essentially, if you just have a single time point, we cannot say much, right? You don't have the temporal information to help you see which regulates which. Yeah, but that's a very good question. Yeah. Okay, so these are, again, challenges. Why is learning U confounder so hard? Because we need you to be pre-perturbation. However, as I said already, we don't have observation of pre-perturbation gene expression or phenotype in perturbed cells, especially if you have a single time point. That's the snapshot you get, right? And as a result, it's a snapshot. Then in perturbed cells, their transcriptomes already mix the basal state and the response to the perturbation. And there are many approaches, as I will name them later. So they try to learn U from the post-perturbation data from a single time point. For example, contrastive VI, SE-CAPE, CPA. These are method names that try to learn the basal state embedding using a machine learning approach from perturbed cells and unperturbed cells jointly. However, since they already use post-perturbation transcriptome information from these perturbed cells, there is a risk of being colliders, or we are having some collider information from that. So I want to say this is reality. The first two bullet points are the reality, but in the theory of doing causal inference, the confounder must be causally upstream of X and Y, and also unaffected by the perturbation. So that is the thing you want to be conditional on. But it seems impossible to learn that from a single time point transcriptomes without assumptions. So I think the gap here is that even though we have many, many computational methods that try to do something similar to what I said, but their assumptions are not so clear, and people are not comparing them on the same ground. I think that's the challenge for the people, especially as a computational researcher. Okay, so then I'll do another round of summary of existing approaches. First, I'll look at how they consider perturbation, X. So these methods just use binary X, use a guide RNA indicator intention to treat. So they're doing something like a differential expression by comparing genes with the guide RNA, cells with the guide RNA to the cells without the guide RNA to see which genes have expression changed. Some of them just totally ignore any confounders on X. They just assume the two groups are comparable. Some consider some technical things to adjust for, which is scepter, which adjusts for the sequencing depth of cells and batches of cells. And these are other methods listed here. So that treats the X as continuous. So they want to learn the perturbation efficiency, or how much the gene X is actually knocked down after perturbation. So in this case, these methods make scale, PS, cell cap. They don't necessarily use the word confounder, but they do consider some factors that will affect the perturbation efficiency, and they want to model that out. So that's why they're not just using X as measure from the UMI count from single-cell RNA-seq, but they do some modeling to get the perturbation efficiency of the target gene. Finally, deep learning approaches. There are many of them, and they want to learn X and U jointly as some latent variables. So they will actually do some disentanglement in their loss function training by separating X and U, so that they hope U just reflects basal state of cells before any treatment, and X as the treatment-specific effect. So, but here, whether there's any danger of having collider risk, that's something that has not been discussed. So putting these methods together, this is a table. I want to argue that if we think about what they're trying to estimate as the final output, it's also different because using binary X, continuous, or latent X, essentially they're defining different estimates. An estimate is a statistical jargon, meaning the thing you want to estimate. If they're estimating different things, then of course their results will be different. And this is another thing I want to raise people's attention. Like even conceptually, they're not doing the same thing, and they have different identification assumptions. So yeah, so that's why we have a very wild field here right now. Yeah.
I just have a quick question about the idea of the intention to treat versus the continuous.
Yeah.
It's not just the binarization, right? Because you could binarize
the continuous,
the continuous output.
Exactly.
It could still be good for being,
right.
more sensitive or something. So it's really just the idea that you don't know what it did.
Exactly.
Okay.
Exactly. Correct. Yeah. Okay, open questions. So I think data is very important, as Tom mentioned as well. We can learn as much as the data tells us, right? So if we want to have better data to help us resolve this challenge, what must we measure to identify you? So I think having more than one time point is important, especially this is also related to the question about when you measure the cells after perturbation, right? If we can have multiple time points to give us more information about the effect of perturbation, maybe that's more informative. Yes.
What do you mean by multiple time points? I mean, the measurement is destructive, right?
Yes, yeah.
And so you have to do it in parallel, but then the given Perturb-seq agent, the guideline is in different cells every time.
Yes.
So what I presented yesterday solves a problem. So you need to grow your cell a little bit after your transduction so that you have a minimal subpopulation of cells with the same guide RNA. And then you need to take aliquots. It's the only solution, right?
Yeah, so sorry I missed your talk yesterday because I just flew in early this morning, but I think I'll be very interested to watch the recording. And I think that's exactly what I tried to say. Basically, you want to measure at different time points after the perturbation, right? After you have the guide RNAs in to see what's happening with the transcriptome.
But you have to guarantee that the same guide RNA species is in the stem, what we call cell family,
have
the same transcriptome.
Yeah. Yeah, so I think for now, a lot of I think most of the Perturb-seq is applied to in vitro cell lines, right? So there is an implicit assumption that the cells in the cell line are more homogeneous. But however, whether we should still consider some cell state, like cell cycle, that may affect it, may act as a confounder. That's the question. Okay, another thing I want to just
Because do you mean you can use granular causality-like method to infer, but this is still not the same population?
Not the same population, yeah. But at least you can have more information about perturbed cells pre-perturbation state, right? Because if you believe the
populations pre-perturbation.
That's right, yeah.
So you still need to match population first.
You still need to match population, but at least you have some transcriptome measurement that is happening before perturbation takes effect. That's the thing you don't currently have with single time point data. Because for a single time point, you have to wait long enough for the perturbation to take effect. Then for the perturbed cells, you don't know their transcriptome before perturbation takes effect.
But are there literature, data that just baseline data for every kind of medium that is they don't have those kind of things to compare?
No, so that's why it's open question, right? So I'm just saying, I'm just doing brainstorming here about what type of data we hope to collect if we want to have better understanding. Yeah, yeah. And okay, I'll just move on because I know we're running short on time. So multi-omics. So how can ATAC-seq help us do this better? Because ATAC-seq is reflecting transcription potential. And also, what kind of experiments can we design to separate U from X and Y, confounder from the two genes? So we can have replicate guides per gene, which is already in some experiments. We have multiple guides per gene. And also, how can we have controls that have cell state variant, but not perturbation? For example, can we synchronize cell cycle stages? Can we do lineage sorting? So this is a way to have to give us better matched cell states. computationally, I want to just end here by saying that sensitivity analysis is a must, but we don't see that much in the current methods yet. So how robust are our inference results, the causal inference results to different choices of X? Because we have already seen X can be the binary guide RNA entering, the continuous knockdown score versus some latent treatment effect. And how sensitive are conclusions to the definition of U? If we use different things as a confounder, how different are the results are? And finally, how can we diagnose collider risk? Because one simple way to consider collider is that if you condition on something you think as a confounder, but you observe a stronger causal effect, maybe this could be a risk. It could be that you are conditioning on a collider by treating that as a confounder. So in this case, can we have some negative cases where you have genes not targeted and you have non-functional guides to check your model? Essentially, if you observe any causal effects in the negative control cells, they must be due to your conditioning on colliders. All right, so my take-home message is that without richer data to help us consider or solidify what confounders are, we're forced to make strong assumptions. Then making the assumptions explicit and making sensitivity analysis transparent are mandatory for analyzing Perturb-seq data. That's just the hope that I can see better. And so some information I talk about today is actually discussed in this mini review paper published in Cell Genomics this year. Okay, so I want to thank my student Qing Yang and my postdoc Si Yuan for helping me prepare this talk to talk about open questions. Thank you. Yeah.
And then we can take questions, but we'll have Ian start setting up.
Sure.
Jessica, there's a
Yeah,
question.
you can take questions. If you
Sure. Okay. Yeah. Oh, should I keep the mic?
You can. Yeah.
Okay. Okay, sounds good. Yeah.
I think Brian.
Yes.
Just to add one further complexity, something you mentioned cell cycle score, like we might like to condition on cell cycle score, but the reality is that the perturbation unfolds over a period of time that is like on the cell, same time scale as an entire cell cycle. So it's not like it's introduced precisely at one time when the cell was in, exact time point of G1 or something like that. It's sort of the effect of the perturbation unfolds over many hours or even days. So some of these things are not, like there are some obvious ones that you could or might like to condition on if you just have a heterogeneous population of cells, maybe you don't want to mix up your.
That's true. Yeah.
Others I think are even a bit more complicated.
Yeah. So that's why whether you can have the cell cycle stage when you perform of the cells when you perform the experiment, right? When you have the guide RNAs and like the entering time of the cell cycle, that's the thing. Because as you said,
when the virus goes in, or
when the virus goes in. Yeah. Or do we have any understanding about when does perturbation take effect? How long does it take to?
Takes a while.
Takes a while, yeah. But that time length also depends on the cell type, right?
Yeah.
Yeah.
I have a question.
Yes.
Just a practical question. So like on your second or third to last slide, you had a list of different methods for doing sort of the same sort of processing.
Yeah.
How different are the final results if you go down these different paths?
Yeah, that needs a benchmark study. We haven't performed that ourselves.
Okay.
Yeah.
Because I mean, from a practical perspective, if I'm a biologist saying which tool do I use?
Exactly.
Does it matter?
Right.
Like, or are we like nitpicking things that the final outcome is essentially identical?
Yeah. So that's why I think benchmarking has become more and more important and essential nowadays for the field, even though it is a lot of work, right? But maybe AI can be helpful in some ways because you can run the software maybe more easily nowadays. Yeah. But still, I think having data sets, so benchmark can happen at multiple levels. One is to check the consistency of these methods outputs, even though you don't know what's the ground truth in the real data. However, the challenge with this argument is that consistency may not indicate they're all correct. Maybe they're biased in a similar way if you make the similar assumptions. So that's why how can we set up the benchmarking standard is another question.